Hello everyone and welcome to Chapter 16, Inherited Change. Now, last semester we have done 4 chapters of A2 so far, and this semester we are going to do 4 more starting with this chapter. Now, what is inherited change all about? In a nutshell, it is about how traits are inherited across different generations. Why is it that you and your siblings may not look exactly the same even though you have the same parents? Traits or phenotype or appearance is determined by 2 things mostly. Number 1 is genes. So we will be doing a lot of genetics in this chapter, and I love genetics, by the way. And number 2 is environment. So environment can also influence how you look like. But we're going to do the environment bit next chapter. So this chapter is all about genes and how it's being controlled and how is it being inherited. Now, there are 4 major parts in this chapter. Number 1 is meiosis. Meiosis is cell division, but it forms haploid cells instead of diploid cells, unlike mitosis. Number 2, Mendelian inheritance, and also your first stats test ever, the chi-squared test. Number 3, we're going to learn what happens when things get more complicated, so more genetics. And number 4, we are going to talk about gene control. Gene control is all about how the genes are expressed, okay? And when they are expressed, what controls gene expression, essentially. Those are the 4 parts. But before we go into the first part today, which is meiosis, we are going to go through some important terms. Okay, some of these are revision, but we're going to add a few new terms here as well. So here we go. Number 1, homologous chromosomes. Homologous chromosomes are a pair of chromosomes. Easy. One is inherited from the father, one is inherited from the mother, okay, usually represented in red and blue like this, but not always. You can tell that they are a pair by the size. They have the same size, they're the same genes. You can see GGRR, okay? They have the same gene loci. I'm going to write here, gene loci. Loci is a position, but they may have different alleles. So this is big G, this might be a small g, or it could be the same as well. So it may have different alleles. It's not for certain. Now, that's homologous chromosomes. Now, chromosomes also have different names depending on what they code for. So there are sex chromosomes, X and Y chromosome, and autosomes. All chromosomes except those sex chromosomes. Okay, sex chromosomes, we have 2, which means autosomes, we have 44, right? Which means how many pairs is that? That is 22 pairs. Okay, 22 sets of homologous chromosomes that are autosomes. So I hope that's a good revision for you. Now let's continue to define a few more things here that you already know, but I really need you to remember this and know it for real. It's going to help later when things get more complicated. So let's define gene. What is gene? A specific DNA sequence that codes for a polypeptide. By now, you should have memorized this sentence already. A specific DNA sequence that codes for a polypeptide. Now, when we say a gene is sex-linked, this means that a gene is on the sex chromosome. There's X-linked genes, which means it's on the X chromosome, and there's also Y-linked genes, which means it is on the Y chromosome. There's also autosomal linked genes, which just means that they are genes on autosomes. Now, if I delete the word in front, taking out sex and autosomal, these 2 words, this becomes a different sentence, a different meaning. When I just say it's a linked genes, and I say that these genes are in a linkage group, I mean that all these genes can be found on the same chromosome. For example, you can say that B and T are found on the same chromosome. Right? Gene B and gene T. This means that they are in a linkage group. they are likely to be inherited together because they're all on the same chromosome. I can even say GRST. These genes can be on the same chromosome and therefore are in a same linkage group. So it's very likely that you inherit GRSBT together from your mom, maybe. Okay, so that's linked genes. Now, the next thing is also familiar to you. Okay, I'm just going to show both of them at once. Allele. Allele is a form of gene. Again, this one you should have memorized by now. It is usually a result of a mutation. So between homologous chromosomes, again, it might be the same gene, same loci, but it would have different alleles. It may have different alleles. It could be the same. It could be different as well. And usually it's a result of mutation. Now, we have learned an example of mutation in sickle cell anemia in AS. Now, as you know that the allele could be just one base different. It could be just a few bases different from each other. And that makes a different allele. Just a few amino acids here and there would make a different allele. And it's a result of mutation at that certain points only. The next word is locus, which I've said a few times in this video so far. This is the position of the gene on the chromosome. Again, you should already know this. Now, I'm going to go through the next slide quite quickly because I think this one you know for sure already. This term is diploid cells and haploid cells. Now, in diploid cells, there is two sets of chromosomes, meaning that they have two copies of each gene and two alleles of each gene. Okay, these alleles may be same or may be different depending. Now, diploid cell, one set of the chromosome is from the maternal side, so one inherit from mom, and the other one is paternal, is inherited from your dad. All somatic cells are diploid. Somatic cells are all cells except for gametes. In humans, somatic cells have 46 chromosomes. This is the diploid number in humans. And usually, this is a result of mitosis. However, there's such thing called haploid cells as well. Haploid cells only have one copy of each gene because it only has one set of chromosomes, and this usually is in gametes, or in other words, germ cells. Same things. Haploid number is obviously half of the diploid number, 23 chromosomes in human beings. Different organisms, different. And this is a result of meiosis. So without further ado, let's go into part 1, meiosis. In this part, we are going to learn the process and different stages that occur in meiosis, how meiosis actually causes genetic variation, and how is meiosis used to form gametes in humans and how to form gametes in plants. So 2 organisms here. Let's start with the importance of meiosis first. Now, meiosis is also called reduction division for obvious reasons, because in meiosis, haploid cells are produced. Okay? So, the function of meiosis is to produce gametes, sex cells that are haploid, which means it has half the number of chromosomes. This occurs before fertilization in sexual reproduction, so in your ovaries or in your testes. Yeah, so this happens before fertilization. At first, we would have 46 chromosomes, as you can see here, it's written here. But after meiosis, the chromosome number is half to 23 chromosomes each. So this is a haploid number. Okay, and this happens in both the mom and dad. After that, the sperm and egg would fertilize or fuse together in fertilization to form a diploid organism again. So the importance of meiosis is also to maintain the diploid number of chromosomes in each generation. Okay, if you don't understand that, why it's phrased this way, why maintain the diploid number, think of it as maths, right? You need to have half from the mom and half from dad in order to make a diploid organisms. What happens if meiosis does not occur, but instead mitosis is used to form gametes? It's going to be crazy. It's a nightmare. Why? Because at first you have 2N. And what is the product of mitosis? 2N, diploid cells. And what happens during fertilization if your gametes were diploid? The offspring will result in 4N. And 4N is a tetraploid. It is not a diploid organism anymore. And it would be just a nightmare. So it's very important that gametes are made via meiosis, and this maintains the diploid number of chromosomes in each generation. Now in mammals, in animals, this rarely happens, okay, tetraploid organisms, and these organisms that are tetraploid, especially human beings, will not survive. In plants though, plants have a superpower, and we'll learn about it next chapter, but plants can be tetraploid sometimes. Anyways, focus. Meiosis, again, to produce gametes, half the number of chromosomes, and to maintain the diploid number of chromosomes in each generation. There's also one more importance that may not come very instinctively until you know the process later on. And that is that meiosis actually produces genetic variation in offspring. What does genetic variation mean? You and your sibling, right? You're not genetically identical. You have differences in appearances, in height, maybe even intelligence and beauty. Who knows? Yeah. But the idea is you have genetic variation. You look different. Your genes are slightly different, even though you have the exact same parents. Now, this process, what makes you different, is because of meiosis, and we'll find out how meiosis does this later on. For now, let's go through a little overview on what meiosis does. Now, there is interphase before meiosis as well, okay? You should bear in mind. And what that does is that DNA replication occurs and this butterfly shape here is formed. We're going to focus very much on what happens in meiosis 1 in general and meiosis 2. Now in meiosis 1, this diploid number is half. So 2N become N. Chromosome number is half. Why and how? That's because the homologous chromosome separate. Very different from mitosis. Yeah, in mitosis, the centromere divides, okay? But in meiosis 1, homologous chromosomes, those pairs, would separate. And in meiosis 2, what happens is that the sister chromatids are the ones that separate. And this is a lot like mitosis. So if you don't remember mitosis at this point, you should, because you just had your AS. Pause this video and go revise mitosis. It will make your life so much easier. Now just a little summary of what I just said. Meiosis 1, very important. Remember that the homologous chromosomes separate first. This results in 2 haploid cells. Okay, but meiosis is not over yet. Meiosis 2, the sister chromatids of each chromosome separate, and this results in 4 haploid daughter cells. At this stage, meiosis is complete. Okay, so what are the stages in meiosis? The good news is it's going to be super easy to remember because it's the same names, except because there's stage 1 and stage 2. It's prophase 1, metaphase 1, anaphase 1, and telophase 1. Okay, that's just a 1 behind. And in meiosis 2, you just add a 2 behind. So prophase 2, metaphase 2, anaphase 2, and telophase 2. And of course, there is cytokinesis in between them for the division of the cytoplasm. Okay, ignore the diagram first. We're gonna get to that right now. So prophase 1. These are the things you already know happens in prophase. Condensation of chromatin to form chromosomes that are visible. Centrosomes move to opposite poles. Nuclear breaks down. Nucleus disappears. Spindle fibers form and such, attaching to the centromere and pulling them to the right position later on. But what is different from the prophase you know and prophase 1 and meiosis 1 is this. Number 1 is synapsis. Repeat it after me. Synapsis. Now, what is synapsis? Synapsis is where the homologous chromosomes, so the pairs, pair up. Okay, they find each other and they come for a date. And each pair is called a bivalent or a tetrad. You can use both these names. It means the same thing. Bivalent as in there are 2 of them. Tetrad as in there are 4 sister chromatids lined up together here. Now, after synapsis occurs, something else occurs as well that makes prophase 1 extra special. And that is crossing over. Okay, and you can see this taking place at the chiasmata. These are the areas where the homologous chromosomes overlap. Okay, and this crossing over is in essence an exchange of genetic material between the non-sister chromatids. So let's take a closer look about what crossing over is, and there are some diagrams here to help us as well. So what happens is during synapsis, which is the pairing of homologous chromosomes, they don't only just pair, they also coil around each other intimately and remain in contact at the chiasma. And it is at the chiasma, this area, or chiasmata, chiasma is the plural, chiasmata is the singular, yep. And this is where those arms are swapped. Basically, part of the chromatid break from its original chromosome and joins to the other non-sister chromatid on the other chromosome. It's very important to use this term non-sister chromatid. It comes out in the mark scheme a lot. Now, this results in a few things. Number 1, we already talked about it, is exchange of genetic material. Now, it's important that in the exam, you do not write exchange of genes because it's not individual genes that are being exchanged, but it's the entire chromatid. So it's exchange of the entire portion of genetic material between non-sister chromatids. Now, the other thing that happens is the linkage group is broken. Now, you can see here, very tiny, but you can see it, that this arm is supposed to have a small a and a big B. Okay, but and this blue one here, it has a big A and a small b. But because of the exchange of genetic material, because of crossing over, instead of a big A and a small a and a big B, you have a small a and a small b. And instead of a big A and a small b, you have a big A and a big B here. These chromosomes have been slightly altered, yeah. The linkage groups, okay, which is small a, big B, which is in a linkage group because they are on the same chromosome together, this is broken now and replaced with a different allele. Therefore, there is a new combination of allele within each chromosome. And this is good because this results in genetic variation. We will go into that in more detail after we finish all these processes. So for now, what's the most important thing for you to know? In prophase 1, other than the stuff you already know, which is here, there's also synapsis, pairing of the homologous chromosome, and crossing over that takes place. Alright, moving on to metaphase I. Now, what is special is that everything is the same, but the homologous chromosomes are lining up in pairs. Okay, so centrosomes reach the opposite poles, spindle fibers are completely formed, it's attached to chromosomes and centromeres, it's the same, but the lining up is not 1 by 1, it's 2 by 2. Bivalents or tetrads line up across the equator or metaphase plate. Now, this is where something we call independent assortment occurs. Fancy name, easy concept. Independent assortment of homologous chromosome pairs means that each pair lines up independently of others on the equator. Now, that's the nice, fancy way of putting it. But basically what it means is that it could be red up, blue down. It could be blue up, red down. It doesn't matter. It could be either way. It doesn't matter. The pairs, or for red, doesn't all line up on one side and blue all on the other side. Doesn't work that way. Okay. So each pair lines up independently of others on the equator. This is going to become very important later on when we talk about genetic variation as well. Independent assortment of homologous chromosomes. And because this results in gametes that are genetically unique. And we'll see why when we see the whole picture. So let's finish this first. What happens in Anaphase I? You guessed it. Since they're lining up 2 by 2, you do not expect the sister chromatids to separate. You don't expect the centromeres to divide. You expect each chromosome to be pulled to opposite poles. The homologous chromosomes are the ones that are separated and pulled to opposite poles. Again, the word poles here is very important. You cannot use ends, you cannot use side poles. Okay. And of course, the centromeres are the one connected to the spindle microfibers. So they are leading towards the poles. You can see here that they are closest to the poles. And in the end, we have telophase I and cytokinesis. Now in telophase I, because the cell is going to go into prophase I again, it's going to partially decondense. The chromosomes will only partially decondense, but everything else is about the same. Yeah, nucleolus reforms, whatever resembles something break down, and cytokinesis occurs to form 2 haploid cells with half the number of chromosomes. Easy. Now after telophase I and cytokinesis, alright, which you should know from chapter 5 here. I've inserted the slides, but I'm not going to go through it in detail. Okay, cleavage for forms in plants is slightly different. Right, after all that cytokinesis, there is a brief interphase between meiosis I and meiosis II. This brief interphase doesn't have any DNA replication. It's just some growth, some synthesis of proteins or other substances. There's not a lot of things going on, and it's called interkinesis or interphase 2, because this is the second time it's going through interphase. Now plant cells don't even go through interphase 2. This is for animal cells only, and in plant cells there's not even the formation of nucleolus and the nuclear envelope. And that's the end of meiosis I as well as interphase 2. Now moving on to prophase 2. Now prophase 2, and as well as the rest of meiosis 2, is just like mitosis. Exactly the same, just that now you're starting off with a haploid cell with sister chromatids, with chromosomes that have sister chromatid. You expect condensation, all this stuff, right? During metaphase, it looks normal now. The chromosomes are lining up the metaphase plate 1 by 1. It is not in pairs anymore, and this is at right angles to the first equator, which is quite interesting. So think if it's if the previous cell has divided this way, then the next cell is going to divide this way, okay, in right angles to the last division, to the first equator. Now chromosomes are attached to spindle fibers and centromere, and the only thing that's special here is that there are independent assortment of sister chromatids. Okay, it could be this way around or it could be flipped. It doesn't matter. Okay, the blue could be on left side, or the blue bit here can be on the right side, depending on the cell. It's sort of random. Yeah, the second chromosome is the same. It could be red bits on this side, or the red bits could be on that side. It could be flipped here or this way or that way. That's the only different part about metaphase 2 compared to mitosis. Now again, anaphase 2, telophase 2, very similar to mitosis. You expect the centromere to divide and the chromatids to be pulled to opposite poles here. And in telophase 2, you expect it to decondense completely for a nucleolus and nuclear envelope to reform, and spindle fibers to break down, and in the end cytokinesis occurs. And the result of all that meiosis II work is to form 4 haploid cells in total. So one cell became 2 cells in meiosis I, then those 2 cells became 4 haploid cells in meiosis II. Just a brief overview, prophase I, special things that happen, synapsis and crossing over occurs, and in metaphase I, it's lining up in pairs. Now, in Anaphase I, those homologous chromosomes separate, and then telophase I happens and Interkinesis happens. The rest of meiosis II is just like mitosis. So just a little bit more extra compared to what you have learned in mitosis. Okay, now that we've done with the process of meiosis in meiosis I and II, let's answer this question. How does meiosis cause genetic variation? If every single child has the same parents, why is it that they look different? Okay, and that's because the gametes result of meiosis are different. What causes this? There are two main things. Number one, crossing over, and number two is independent assortment. So number one, let's go through it again. Crossing over at prophase I. This is between non-sister chromatids of homologous chromosomes, and this happens at the chiasma. Now the result of crossing over, obviously, as we have gone through just now already, is exchange of genetic material, not genes. The linkage groups are broken as a result. There's new combination of alleles, and the result is genetic variation because each gamete would have a different combination of allele as well. And number two, as we mentioned just now, is independent assortment. Okay, majorly, okay, it is a major reason of genetic variation, and this happens at metaphase I. This here is an independent assortment of homologous chromosomes, where each pair lines up independently of other chromosomes on the equator. So you can see here there are two possibilities of arrangements. Now, this cell here only has 4 chromosomes, and there's 2 possibilities of how it could look like. But imagine if you have like 46 chromosomes. So if you have 46 chromosomes, your N would be 23. So the possible number of combinations would be 2 to the power of 23. There is a lot of different combinations that we could end up with. A great cause of genetic variation. This results, obviously, in gametes as well that are genetically unique. You can see here, if it's arranged this certain way, the gametes will look like this, and if it's possibly 2, then it will look colorful a little bit like that. Now there's another kind of independent assortment, and this is independent assortment of the chromatids. But this is not so important, okay? You realize that there's a lot of words here, but less words here, and that's because this contributes to genetic variation a lot less. Why? Because, well, it depends if there is a area where crossing over has occurred. But crossing over doesn't happen on every chromosome. In fact, crossing over might be a relatively rare event. Okay, it doesn't happen all the time. It's not that often, only sometimes. But if it does happen, and then at metaphase II, the chromosomes are arranged differently, this will also result in different gametes that you can see here. But again, if they ask you why meiosis causes genetic variation, your answer should be number one, crossing over, number two, independent assortment of homologous chromosomes, not chromatids, that happens at metaphase I. Okay, this is a side point. Now there is one more point, which is also a rare occurrence, but it can happen, is that there might be some possible chromosomal mutation. Now we're not talking about gene mutation, which is just like one base or two base that usually happens during DNA replication. We are talking about chromosome mutation, as in the whole chromosome structure or the chromosome number changes, resulting in new alleles or even new diseases. Now, this is a famous example here, which is Down syndrome. It's also called trisomy 21. We'll spell it out for you. And that's because they have 3 chromosome 21 due to a meiosis gone wrong. Now, that's not desirable, obviously, but it happens. So that's how meiosis causes genetic variation. May cause genetic variation. But hey, besides meiosis, what causes you and your sibling to look different may also be due to fertilization. Okay? Well, you look at the reasons and you'll laugh, okay? And this is due to, number one, random mating, and number two, random fusion of gametes. Humans are monogamous and they usually stick with a partner for very long periods of time. But imagine small guppy fish which mate non-stop with random other fish, right? That would cause a lot of genetic variation in their offspring as well. In addition to that, sperm and ovum. There are millions of sperm, maybe a few ovums, and which sperm gets which ovum. There is an element of chance as well, right there, that could cause genetic variation.